Welcome to another TPD seminar. We have two really great speakers today. We're switching focus a bit and looking at the power of AI in TPD. So our first speaker today is Luca Naef. He is the co-founder and CTO of VantAI, a biotech focused on an AI-first approach at unlocking the potential of induced proximity. VantAI is a trusted partner to and has achieved multiple preclinical milestones in several major pharmaceutical and biotechs in the protein degradation field, successfully applying AI across a range of novel effectors and difficult-to-drug targets. Luca received his bachelor's and master's and significant research experience across ETH Zurich, Stanford, and other global institutions, and has worked extensively in the AI field, both as a researcher, software engineer, and devising and implementing the AI strategy of global pharmaceutical companies as part of McKinsey and Company's Quantum Black Unit. So I'm going to hand it over to Luca for our first talk.
Cool, amazing. Thanks a lot, Catherine. And I'm really excited to talk to you today about current and future approaches of accelerating induced proximity through AI. And as an avid watcher of this series, it's a pleasure to share some of the work that we've been doing at VantAI of accelerating this field. And I'm going to kick off with a quick introduction about what VantAI is all about. We really position ourselves at the intersection of what we believe are three major phase-shifting technologies. Number one, induced proximity, so targeted protein degradation, but also going beyond that. Number two, a flavor of deep learning called geometric deep learning. And number three, structural proteomics, which really allows us to generate the large amount of structural data that we need to train those models, but also as a screening platform. And we really got excited about this field roughly 3 to 4 years ago when we realized if you can build the technology to bring any two proteins together, you can convert the proteome into this beautiful biological warehouse of tools. And of course, we're all familiar with ubiquitination, but there's so much more. And obviously, targeted protein degradation is an extensively explored tool in the toolbox. But despite all this work, rational design is incredibly difficult. And it's because not only do you need to design a molecule that binds just right to the protein of interest that you want to degrade, but you need to design the entire complex just right to induce polyubiquitination and subsequent degradation. And so given that, and we have very limited data, you might think, okay, this is a hopeless cause. We're never going to be able to apply computational approaches to this. And I really hope that I can convince you from the opposite today, because while it sounds intractable, it's also uniquely suited for AI. And this is the problem we are trying to solve: bring two proteins together. We introduce a small molecule that binds to one of the two sides and changes the surface of the protein into this neoprotein. This neoprotein has a neosurface and can engage into neo-interactions. This is obviously heavily inspired by Stuart Schreiber's fantastic talk on this very series in 2021. Now, this makes this problem very, very different from classical small molecule drug discovery because it's all about neoprotein design. And that means AI can actually learn from nature to design these neoproteins. So from natural interfaces, for example, Degron-induced substrate interactions, from all sorts of other different geometries of interactions, to mutation and PTM-induced interfaces, to homologous interfaces where the proteins globally might look entirely different from what we're trying to induce, but the solution locally looks just like the neo-interface that we're trying to induce. And then to design those rationally, it's really a geometric puzzle. Can we find just the right shape in electrostatics to find a molecule that designs this neoprotein and engages with the right neosurface? And of course, this sounds very abstract. And so I want to illustrate this with a beautiful example from Vincenzo D'Angioletta's recent talk at the TPD series, where they stipulate that UM171 actually imitates a KBTBD4 mutation induced neo-substrate degradation. And they stipulate that UM171 basically imitates the surface change induced by this mutation, leading to the neo-substrate degradation of CoREST. And the whole idea is that if you can train models to learn these sort of situations from nature, we can approach the design of molecules such as UM171 in a rational way. And to solve this geometric puzzle, the flavor of deep learning that we apply is called geometric deep learning. And this was coined by Michael Bronstein in 2015, our chief scientist. And it's all about deriving architectures for geometric objects by respecting their underlying symmetries. Let me give you an example. If you have a perfectly round ball, you rotate it by 30 degrees. The ball looks exactly like before the rotation. So it is what is called invariant under rotation. So different geometric objects have different symmetries. And if you design machine learning architectures that respect those symmetries, they become much more accurate and much more data efficient. And we really apply geometric deep learning across the whole spectrum. Susanna Kozinski explained beautifully how if you have more extensive protein-protein interfaces, the SER becomes easier and we can design more diverse molecules. So we look for the most compatible effector target pairs. We try to predict their interfaces, identify pockets, identify solutions to the puzzle by identifying ligands and optimizing those so we get drug-like molecules. And of course, this is an academic series, and so what better way to share some of the breakthroughs of applying geometric deep learning in this field than sharing some of our recent and upcoming publications. And we're really committed to open science, so all of these are either already openly available, you can use them in your work, or they're going to become available very, very soon. And so we're going to kick off with interface prediction, where we recently proposed DiffMassive, and it's a protein surface diffusion model for interface prediction. And it really brings together two key components. Number one, protein surface representations, and number two, a new class of models called diffusion models. And we really got excited about protein surface representations once we started thinking about neoprotein design from the lens that I just walked through, because if we can learn from all naturally occurring protein surfaces, we can learn how to design neosurfaces. And this line of work really started around 2019, which is roughly when VantAI also got started, with the first methods introduced that learn on protein meshes, a method called molecular surface interaction fingerprints, or MASSIF. And this was then followed up in 2020 by this first end-to-end differentiable work that uses protein surface point cloud and learns a quasi-geodesic convolution that respects the local geometry of the protein surface to perform this very efficient network updates. And this work was led by Freya and John. And all this line of research was actually pioneered by Michael and Bruno. And it's obviously a pleasure to have both of them on board with VantAI now. And they've also recently published this beautiful work in Nature, where they used their predecessor, MASSIF, which uses meshes, to design extremely potent SARS-CoV-2 receptor binding domain binders that were using these protein surface fingerprints. So this is from Michael's and Bruno's lab as well. So we got really excited. It's a really nice fit, and obviously this works fantastically well for protein design. And so we really wanted to combine it with this second key piece, which are diffusion models. And if you have not been living under a rock, you might have realized that something happened in 2020, where all of a sudden we're able to feed tools like Midjourney with prompts like show a molecular glue holding together two planets, and this is what we get out. Okay, we added the glue in post-production, but the planets are real, and this is fantastic. These are photorealistic images, and such a big jump from what we were able to do just a few years ago. And what's behind this is a model called denoising diffusion probabilistic models, or short diffusion models. And they're inspired by physical diffusion processes, such as you might find them in Brownian motion. And instead of diffusing over motion, you actually diffuse over, for example, ground truth images by adding noise to them in a stepwise fashion. And I'm going to go into a lot of detail there now. Now, there's a tradition to explain this using CAD images, and I'm not going to break that tradition. So imagine you have millions of CAD images, and you start with the original ground truth image on the left here at time x0. And during training for all of those CAD images, you successively add noise to image until you added a completely noised image at xt capital. This is called the forward diffusion process, and you can calculate this analytically. In the reverse diffusion process, and this is learned by your model predicts, given the noise and the time step t, what noise was added in the previous step. You can run this for all time steps t until you ideally end up at the original CAD image. Now, this doesn't sound very useful, but you can actually use it at inference time once you've trained this on millions of images to feed pure noise, and it can generate these photorealistic images. Now, cats are awesome, but we're a lot more excited about protein complexes. So the way that you can train these sort of models on, for example, interfaces, both from ternary or binary complexes, is that you feed it in thousands and thousands of examples of complexes. And here we're not diffusing over pixel space, but we're actually diffusing over rotations and translations of the pink protein. You can see it gets rotated, translated until it ends up at this completely jumbled position on the right. And then the model learns to reverse that. So starting with completely jumbled positions, be able to predict the final pose. And if you train this on many thousands of complexes, you can actually feed it in with completely novel structures that will be able to predict their binary complex. And so really these two pieces together, molecular surface interaction fingerprints for protein surface representations and diffusion models, if you put those two together, you end up at DiffMassive. And this is the method that we introduced, and it was fantastic to be able to work with Freire and John and Michael and Bruno again in doing this, which obviously have pioneered this line of work. And this model converts proteins into protein surface point clouds. It has an equivariant encoder decoder architecture, and then, like we talked about, predicts rotation translation starting from completely jumbled to the final truth position. And what's really nice is that we also add this auxiliary task of site prediction. So not only do you have very interpretable insight where the model thinks the final proteins will end up, but you can also feed it information that you have. So you might have, for example, HGX data on your ternary complex, and you can feed this in really nicely into the predictions to make them more accurate. And here you can see that this actually works tremendously well. So you can see a comparison against some of the other state-of-the-art machine learning-based docking methods, and you can see our method is able to outperform previous diffusion and non-diffusion-based methods quite a bit. And this is really nice, but the main thing that we wanted to achieve here is get away from relying on coevolution. And most of these methods, such as diffdoc and methods such as AlphaFold multimer, they rely on coevolution by either being fed directly multiple sequence alignments or by being trained on large sequence databases to learn the coevolution. And if you know how two residues coevolve over time, this gives you a lot of insight on where the potential interface forms. And obviously, this makes the problem a lot easier, makes it more accurate. But if you don't have that coevolutionary information available, for example, for neoproteins, induced interactions, then this is not going to work. And what you typically see, and you actually see the exact same sort of graph for AlphaFold, for protein folding as well, as the coevolutionary information quantified by effective MSA depth goes down, the performance of these sequence representation leveraging models goes down as well. But our method is actually able to perform even in low sequence conservation depth, really showing that we're able to learn the geometric complementarity of these surfaces as well. Now, of course, rigid body docking is nice, but we all know proteins move. And let's just imagine you want to bring CDK12 and DDB1 together rationally. And you just take the crystal structures, APO unbound DDB1, you do docking. If you perfectly nail the interface, you end up with the blue protein here. You can see this is the overlaid CR8, a molecular glue from the 683 crystal from the holo structure. We would never be able to find this molecule if we just docked with the APO structure, because you can see that the molecule clashes because there's an APO-holo conformational shift that is induced by the protein. And so this is very important to take into account. And obviously, you can do molecular dynamics, but that's very expensive. And you also get trapped into these local minimas, the kinetic traps. And so our next line of work focuses on being able to predict those pocket dynamics using a diffusion model called a polydiff. And this models APO to holo conformational protein pocket dynamics using diffusion. You can see the process happening on the right. This is the conformational change we want to move. This is what the model gets fed as an input, and it's trying to predict this one. So the small molecule induced conformational change. And here you can see some detailed network architecture. Basically, what we're doing here, instead of rotation and diffusing over rotations and translations, we're actually diffusing over holo coordinates. So we give it the XYZ positions of C alpha atoms of the holo position. And it starts at, given the APO sequence information and APO structure information, it starts at completely random positions and then diffuses from this random starting point, like the randomly noised CAD image, to the ground truth. And that's what you can see here live in action. So in pink, you can see alpha coordinates start completely random, conditioned on the green structure here, the APO. We're able to recover this alternative conformation using this diffusion model really quite well. So the next line of work that I want to share is how you can drive from initial hits to drug-like molecules during lead optimization using another work that we've published called Shape Linker. And this is as well surface point cloud based, but actually here we're working on small molecule point clouds. And this is for situations where you already have sort of a solution to the puzzle, and you can use a co-crystal structure that you might have available that you want to imitate, or from ternary complex prediction methods, as we're going to talk about in a second. You can extract the solution puzzle piece. And then we have a generative method, an LLM-based autoregressive generator that proposes novel suggestions. We can calculate their puzzle piece shape. We can compare it against what we know solves the geometric puzzle and score the molecule that was generated by the generator via their alignment. And the nice thing is, if you use reinforcement learning, you can actually do multi-parameter optimization, because you can combine the score function with things like rotatable bonds and linker lengths to make the linkers more rigid. and shorter, as is typical during lead optimization. And you can see our method is not only able to match the geometric puzzle piece a lot better than other methods that don't directly include 3D information, but we're also able to do multi-parameter optimization, reduce the number of rotatable bonds and the branching much better than methods that only do 3D design without the multi-parameter optimization. And so here you can see, just as a visualization, where we've designed more rigid molecules. So those aren't necessarily super nice, but they're just more rigid. So we've designed more rigid molecules against some of those previously crystallized structures. And you can see, even though the molecules that we've designed are much more rigid, they're able to match the shape of their reference basically perfectly and ideally induce the same ternary complex. So this can really help you during lead optimization if you have a compound and you want to optimize it. So this is just a tip of the spear. We have many more published works, and we're really excited to engage with the community. So please try these things and give us feedback. But of course, our entire platform combines not only these tools, but much more components. So it's really a combination between large proprietary data sets. I think we have some of the largest induced proximity data sets in the industry, over 70 million degradan peptide E3 ligase affinity data points that we can use to predict protein-protein interaction affinities, over 50,000 proprietary structurally resolved interfaces from our structural proteomics efforts, and a large amount of degradation data points from our internal programs and partner programs that we use to predict degradation models. And this is fed into sort of the successes of some of the initial work that you saw today. And of course, you can't just take these AI-generated outputs, like you can't take GPT-4 just directly and throw it in an email. That would be very bad. You need to refine those answers. And so we also have a really big physics-based refinement pipeline where we have a large-scale high-performance simulations cluster where we take the AI predicted outputs and we refine them. And of course, it's also extremely important to include experimental data. So we usually perform all of these operations in a tight loop with experimental data, and all of our methods can include experimental information that we might have available. So obviously, the first thing that you should do if you design ternary complex multi-pipeline is see, can you reproduce some of those previously crystallized crystal structures? And that's exactly what we do here. So we start from unbound proteins that are available from the PDB, so not the bound structures in the ternary complex crystals, and the molecular structure of the molecule in the SMILE string, so no 3D information. And we predict the ternary complex, and then we compare it against what has been crystallized. So could we have been able to predict this? And you can see 70% of our predictions reach within 10 angstrom of the truly crystallized crystal protein. This is more than double than some of those previously published methods. And you can see here, this really goes across all these systems that we were able to compare. We're able to significantly improve performance. And even for cases where we might not be able to have a perfect hit rate, you can see we're still able to produce in pink here these really crystal-like poses for both the molecule and the protein pose. And here's just some more visualizations. Again, in pink is what we predicted, and in blue, you can see the reference, and you can really see how we're able to produce crystal-like poses for almost all of these published ternary complexes. Now, we've done the exact same with molecular glues. Here, there's less reference methods available, so we just used one of the state-of-the-art protein-protein docking method, and we looked at the protein-protein part. And here you can visualize one of the points I was talking before. Methods that rely on coevolution, such as AlphaFold multimer, and I assume their successor as well, they're not able to predict well on these induced interfaces. We look both at degrading glues and non-degrading glues, whereas we're able to produce highly accurate protein-protein docking poses across many of those systems. Again, here a per system breakdown. And again, to further my point, the only two systems where AlphaFold multimer works well are cases with homodimers or where there's a natural interface with some coevolutionary information available, whereas we're able to really consistently produce highly accurate poses. And again, some visuals that can show in pink what we predicted, 2 different shades, and both for the molecule and for the protein, we're really able to produce these highly accurate poses. Now, of course, benchmarking is important to show that it works, and publications are nice. But at the end of the day, what matters is, can you actually have real life impact with these methods? And so I want to show some case examples. First, target effector selection. And so typically what we do is we combine both the big systems biology component where we look at things like expression and E3-POI symmetry, and a big structural component where we run some of those models, for example, that you've seen previously, and we stack rank all possible E3s such we get the perfect match for a target. And here you can see some experimental validation. We prioritize a completely novel E3, identifying the binder, and then put it into a pan-kinase cell viability assay. And you can see this is the first iteration. We've got much more optimized and clean data on those compounds later, but you can see at this first iteration, we were using the novel E3 that we found. We're able to induce UPS and CRL-dependent cell killing, and this is confirmed by netylation, inhibitor, and anchor and warhead controls. We're also able to validate our ability to predict ternary complexes. So this is a fantastic collaboration with Blueprint and Proteovant, never previously degraded kinase. We prospectively designed compounds using our ternary complex modeling pipeline. We were able to successfully degrade this target for the first time, and luckily we got crystal structures. And so these are unpublished crystal structures. You can see the predictions that we used for the molecular design are within 5 angstrom of the crystal structure that we were able to resolve, really showing that there's probably just a low energy pose that the molecule actually assumes. We can do this in a real life setting. And we can also do this to target a lot of new targets. So this is a fantastic collaboration with Dermaxon, previously never degraded atypical kinase immunology target. This was pre-AlphaFold 2, so we've been on this for a while. And we had to use homology models, and despite this noisy input, in this first round of compounds, 7 out of the 8 compounds were able to grade this never before degraded target on first try in a ridiculous 85% hit rate. Obviously, this is a little bit cherry picked. We don't get the same every time, but we've done this on many, many projects successfully. And it also induces the phenotypic response you would expect. And we've also done this on much, much larger scale. So this is during lead optimization, again, Blueprint Proteovant collaboration. And what we usually do here is once we have a couple of compounds with degradation values is we actually try to predict degradation directly based on the predicted ternary complex structure. And that's what you can see here. We've done this both for the target and for the anti-target. On this large-scale lead optimization program with difficult selectivity of an oncology target, over 100 compounds eventually that we use to train them all. And you can see these are completely unseen compounds, but yet the model is able to predict degradation quite accurately. And I don't have time for many more case studies, but I can just tell you that we've done this across many projects where we've literally synthesized thousands of molecules, and this is very typical of what we can see. Cool, amazing. So quick summary. I hope I've been able to convince you a little bit that rationally induced proximity is all about small molecule-driven neoprotein design. And that means that since we can learn from all proteins in nature, AI will actually have a disproportionate impact in this field in particular. The last 2 years has seen significant breakthroughs in geometric deep learning and generative models, such as protein surface representations and diffusion models. And we've been lucky to be at the forefront on some of them. And AI is in the future, but it actually drives real-life impact today across every single step. But the title of the talk is Current and Future Approaches. And my time is nearly up, and I've only talked about current approaches. So what do we believe the future is? Well, we think it'll require more data, a lot more data. And so for 2024, we're committed to generate up to 1 million structurally resolved interface data points through our structural proteomics platform, which depending on how and who counts, is somewhere between 1 and 3 orders of magnitude more than what we have now. And as I said, we're big proponents of open science. So we also actually recently, or we're going to release next week at the MOMO conference, a community data set that should push the field forward, which has more than 10 times as many PPIs than the currently largest state-of-the-art data sets.